Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with not one, but two amazing guests, Sonya Dumanis and Andy Singleton. Andy is the program lead for GP2, which is a remarkable program in both scale and depth that's tackling Parkinson's disease. And as we'll talk about in the show, I think also has implications for team science across genetics and precision medicine more broadly, not to mention within neurology. And Sonya is with us as Chief Operating Officer of the Coalition for Aligning Science, where she oversees and is part of a pretty significant portfolio there. But for our focus today, it's part of a program of work called Aligning Science Across Parkinson's, or the beautifully named ASAP, which oversees the GP2 program. So we will work through that for everybody to give you a sense of these different initiatives, how they fit together, and what they could do for the field. And maybe, Andy, I'll start with you on that. If you could just give people a sense of GPT-2, who it's for, what the aims of the program are, and give people a little bit of sense of the scope and scale, that would be great.
Sure, Patrick. Well, first of all, thanks for having us here today. It's fun to talk about this project. Hopefully, one of the things you'll take away is how enthusiastic we are about what we're doing and how excited we are about the work we're involved in. So GP2 is the Global Parkinson's Genetics Program. I co-lead this along with someone called Cornelis Blauendraut. And it has a very, very simple aim, which is to dramatically accelerate our understanding of the genetics of Parkinson's disease and related disorders, but to do so in a global context, right? Parkinson's is a global disease. We need to understand the disease across all populations. In terms of product, GP2 is a 10-year program. We're about halfway through the program right now, that aims to generate data on more than 250,000 people from around the world. And that's a mix of whole genome sequencing and genotype data. And to use that data and make that data available to researchers around the world to accelerate understanding. We were actually the first resource program supported by ASAP, and I'm sure Sonya would love to tell you a little bit about ASAP.
Yeah, I think that's a perfect segue. Sonya, talk a little bit about the broader ASAP umbrella.
Sure, absolutely. So ASAP, Aligning Science Across Parkinson's, is a research initiative designed to drive new ideas into the clinical pipeline for the Parkinson's space. Our mission is to facilitate collaboration, to create research-enabling resources, and to also facilitate data sharing in order to accelerate our understanding for Parkinson's disease and inform the path to a cure. The way that we go about with ASAP is we have resource programs, which are essentially core programs brought together to work with researchers around the world, but we also have team science programs in our collaborative research network as well. And so when we were thinking about ASAP in the early days, so Kemney Riley is the managing director there, and then Randy Sheckman is spearheaded and acted as the scientific director. He's a Nobel laureate who in 2017 unfortunately lost his wife to Parkinson's disease. So the two of them teamed up and essentially thought, what could we do that's transformational in the space? They did a huge whirlwind listening tour, and on this listening tour, that's where they met Andy and asked Andy, if you could do something that's game-changing in the space, what would you do that serves an unmet need and really amplifies and coordinates efforts around the world? And that's how GP2 came into being.
And what did you say at the time, Andy? Were you at the NIH at the time? Maybe you could just take us back to that moment. What was the field like then? And when you sat down on that whiteboard or piece of paper or whatever it was, what was the opportunity that you saw from that conversation?
Yeah, maybe I'll go back even a little further than that. So genetics had evolved incredibly, even during the time that I'd been involved. So it went from in the late 90s and early 2000s, from a space where genetics was super competitive, right? You had individual labs working on chasing genes, usually in competition with every other lab around the world. And then in the kind of 2006, 2007 era, we started to realize that as we were producing data at scale, that really the way to make discovery was to work together and to collaborate. So we had formed a consortium back in 2009 of investigators from around the world, although mostly from Northern Europe and North America, to bring together genetics data in the neurodegenerative field, particularly in Parkinson's disease. And this was great, right? This was a coalition of the willing, really, people sharing data, working freely, collaborating really effectively. But we didn't really have much support. So we had this willingness to drive genetics forward. We kind of knew how to do it. I think we knew how to scale things, but we didn't really have a lot of foundational support. And then this opportunity in 2018, 2019 came along, where the folks from ASAP said, we want to do something. We want to do something that's impactful, where you shouldn't worry too much about scale. You should just do what needs to be done. You should also not worry about the details of how you get there, but rather have the flexibility and ability to change plans, to react to changing circumstances, to react to new opportunities. So this was just an absolute dream. You can imagine it's a dream, right? It's something that answered everything that we'd been looking to try and address. So it immediately made me think about, how do we bring geneticists together? How do we tackle the critical questions? And I think for all of us in the field, a question that was burning in our minds was around understanding the genetic basis of complex diseases in populations beyond those of Northern European ancestry. So that's kind of where the dream started.
How much progress have we made on answering that question to date? My understanding of the way that GP2 works is you've built a very large consortium of partners that are agreeing to the data sharing paradigms and coming together to create something that's much bigger than anybody can do in isolation. Where are we today in terms of solving that representation gap, and what's the gap that still needs to be closed, just to help people understand the current state?
Yeah, so there's still a gap for sure, right? I mean, we have 20 years of producing data in Northern European ancestry populations, and really only a concerted effort outside of that for the last 4 or 5 years. So there's a gap, but the gap's closing. And for many of the populations that we work in, if you'd looked 5 years ago, there was no data, there was no genetic data. So one of the really neat success stories is a study from West Africa, from Nigeria, Lagos in Nigeria. And, I think there were 50 or 60 people with Parkinson's who'd been genotyped 5 years ago. Now we have several thousand, and we're making discoveries. So the gap's closing. There's plenty more to do for sure, but we're also accelerating fairly quickly. So I feel like the playing field is leveling out.
Yeah. Sonya, Andy painted this amazing picture for any scientist of, it's the opposite of the grant that you set in stone and you have to do exactly what the grant says, into more of this, let's take a moonshot, let's do something big, and recognize that we're going to learn every 3 months, every 6 months, every 5 years sometimes, and may have to adjust dramatically how we get there, but the mission remains the same. How do you think about setting a structure up, both from a funding perspective, but also from an organizational perspective, to achieve those kind of goals? Because it's not always how science is done, especially at this kind of large scale.
Yeah, so we were in this amazing space since the Sergey Brin Family Foundation said they were willing to do this. So we knew that we had resources at scale to be able to think about a 10-year horizon, which I don't think a lot of funding
Yeah.
agencies do, or they sort of say at 2 or 3 years, we're not even sure if a budget is coming. So we had the budget and resources, which allowed a lot of that flexibility, but that didn't mean that we wanted a free for all. And so essentially, we think about it in chunks where there's a continual evaluation process to figure out, is this working? Do we need to pivot? How can we make this better? And there have been points around the way. So for example, GP2 started as focusing just on Parkinson's disease. And then a few years in, we realized that Parkinson's disease is really on a spectrum, and we need to think about Parkinson's disease in the continuum of other diseases. And so GP2 made the case for the remit to be expanded and focused on. And so it's really just about making sure that we keep the North Star aligned. And then as that's aligned, never losing our edge and never thinking that we're perfect, and hearing from the community over and over. So Andy can talk about a lot of the lessons learned along the way of how we've shifted our operations based on feedback that we've gotten from others.
Yeah, I would just add to that. I think one of the things that's really allowed us to be flexible is that there's a lot of trust between us and the funders. So ASAP is the funder, and then the implementation partner is the Michael J. Fox Foundation. And I really do view it as a partnership between all of those entities. So we have very clear lines of communication. We work through problems together. We come up with solutions together. We decide, should we be focusing on this opportunity? Should we be course correcting in this space? So that's been fun, right? I think traditionally we get money and then we worry that the funders are going to interfere with what we're doing. And this hasn't been it's been a completely different relationship, and one that I've really enjoyed in this case.
What I was going to try to do is break down the way that I see GP2 working today, and then I'd love to hear from you. What were the challenges along the way to achieving that or continuing to achieve it? I think you're in 60 different countries or close to that, and you've got sites and local partners that are seeing patients typically in a research context, probably if they're involved deeply in genetics. And what you're having to do is work together to do data harmonization, pooled access to these data sets under global research agreements. I can already see the deep logistical and operational challenges of how you do that harmonization, and how you just set up the legal frameworks, maybe even of sharing that data across so many countries. But once you achieve that, like, the scientist in me says that that's an amazing resource that then you can understand differences in global diversity of genetics that drive the disease. Am I right in one of the initial hurdles just being like, how do we get people from data from 60 different countries to fit together in a harmonized way, and then people from 60 different countries to work together around a common vision? What else were the early hurdles that were challenging here?
Yeah, you're right. I would say the other challenge is that we started this in January 2020, and
Yeah.
in March 2020, COVID, right? So a global initiative followed by a global pandemic. In some ways, though, that gave us a beat to kind of pause and really focus on operational challenges. So this was the other component that I really valued in terms of our partnership with ASAP, which is that there was a long-term vision here, right? We had 10 years or so. So it means that we could really take the time to set things up correctly and spend a lot of time working with groups around the world and building trust with those groups. And that's essential, I think, to get buy-in, to get genuine buy-in, and to move us away from this space of helicopter science where, a well-funded scientist flies in, grabs samples, and then leaves. That's the opposite of what we were trying to build here. So having time to do that, I think, was really beneficial for us.
Yeah, one of the aspects of GP2 that is so empowering is that in addition to thinking about data harmonization and data analysis, it's thinking about local capacity. So that way we're a global initiative, but we're growing local capacity within the country. And that could be training to get bioinformatics. And we have training programs for PhDs and master students in underrepresented areas, but it's also building infrastructure. So for example, we supported the first biobank in Peru. We're working on ensuring that there's a good DNA extraction facility in Nigeria. And that means that even with GP2 into play, if other people want to come and leverage that infrastructure, it already exists and they don't need to reinvent the wheel. And so that's what I want to highlight. When we're thinking about long-term vision, we're thinking about how do we build a legacy that lasts, that others can leverage too. Otherwise, if we're just doing it just for ourselves, it defeats the purpose.
We're excited to share that the Genetics Podcast will be recording live at the American Society of Human Genetics annual meeting this year. Join us on Tuesday, October 15th from 12 to 1 p.m. in room 151A for a special conversation with expert guests from academia and biotech on the future of genomics. Lunch will be provided and seating is first come, first serve. We'd love to see you in the audience as we record live at ASHG 2025.
Yeah, I love that, and it allows you. I'm gonna come back to this later, but I want to understand what after you've done all this, people can learn from this and apply it in other cases. But I want to just dwell in Parkinson's and neurology for a little bit here to also pick up on that insight about the spectrum. I've seen in in neurology conferences before this axis between Parkinsonian phenotypes and dementias and motor neuron diseases like ALS, and it's clear that there's these are there's something complex going on where brains become unhealthy in some core and fundamental ways, but then they also diverge in some well-known ways, and I certainly don't have all the answers. It sounds like as a field we don't as well. I'd love to just hear a little bit more about what we've learned about that concept of how much we can learn about neurological conditions more broadly from this kind of model and extending it out beyond Parkinson's.
Yeah, it's a great question, and I can't say that we have all of the answers. I do think a critical component to getting those answers, though, is being able to compare and contrast data across disease entities, but also being a bit more open-minded in how we define diseases, right? So you think about Parkinson's disease, the clinical entity of Parkinson's disease has really been formed from looking in Northern European populations, right? We don't know that the same mechanistic drivers might lead to something slightly different in other populations. And certainly we see that diseases like progressive supranuclear palsy or multiple system atrophy seem to occur at different rates in different countries. So what's the driver of that? In order to be able to understand the differences and the similarities across these diseases, we obviously need harmonized data that's comparable across different sites. And this is a major core component of GP2, standardization and harmonization.
Sonya, from your perspective, how much do you have to rethink the core model of this to extend into other conditions? Because in some countries, these patients will be seen by the same centers, and others it's very different kind of structure, and obviously the data types that you're thinking about collecting and harmonizing and the capacity building, you maybe just increase the order of complexity significantly by expanding it. I love if you could talk a little bit about the balance between expanding that scope and the focus that you get from saying, okay, we're going to be the best global Parkinson's resource, for example, and staying focused on that original mission.
Yeah, absolutely. We're focused on Parkinson's disease. That was the remit of ASAP, and that hasn't changed. But we also want to make sure that we're understanding Parkinson's disease in the context of other neurodegenerative diseases. So what we started doing, and it's really a testament to the GP2 team. So GP2 is made up of a lot of volunteers who devote way too much time to the effort. And they also are pitching and thinking about what do their cohorts have to offer. And so essentially there was this inventory done to say, I know that you see Parkinson's patients, but who else do you see in your clinic? What other types of data do you collect? So we can start thinking about what is low-hanging fruit that we can start expanding and growing. And I often believe in piloting. So let's just test this out. If this works, then we can grow, but take a phased approach. So we're not going to tackle everything all at once, but that doesn't mean that we can't slowly start to try and navigate that as well.
Yeah. I want to talk a little bit about the therapeutics of the future and where we see this going, because this is, for me at least, based on reading the paper that you all wrote recently, but also understanding the program. What we're trying to do here is understand fundamental disease biology so we can develop therapeutics that work for everybody and not just for the populations that have been studied. I would love if you could help, maybe this is one for you, Andy, break down what we know today about genetically targeted therapies in this condition, because listeners of this podcast will know about LRRK2 and GBA and some of the more common genetic subtypes. But what they may not know is what we're starting to understand about polygenic background and how that influences risk and also potential routes of treatment for patients. I love if you could just break down what we know today and what we hope to know as I think you've got about 80,000 sequenced individuals that are available through the platform now, and that will grow to the 250,000 and potentially more through the life of the program. But what might we be able to do at the end of this program once we can really more fully understand the spectrum of monogenic but also polygenic drivers of disease?
Yeah, sure. So again, most of the knowledge to date has been garnered in populations of Northern European ancestry. That's changing because of GP2, but it is certainly changing. What we know is that there are a dozen or so genes that contain mutations that are fairly highly penetrant. And there are around 150 or so risk loci for Parkinson's disease. So you can start to create polygenic risk scores that can, place people in different terciles of risk. From a therapeutic perspective, there are obviously a few things that we think about from the genetic side. The first one is coming up with rational points for therapeutic intervention, right? Mechanistic based points of therapeutic intervention. And this is why many of us started in genetics. I think we're now also starting to think about how to match the patient to the particular mechanistic therapeutic that you're coming up with, right? Is there a mitochondrial phenotype in Parkinson's disease? Is there a lysosomal phenotype? Can we start to say which patient has which flavor and which mechanism we should go after? I think the more recent area that we're really excited about now is starting to think about the temporal sequence of mechanism. So is there a particular mechanism that I would go after early in the disease, after a certain milestone has been reached? And I think genetics is useful there too. So when you bring all of these things together, it comes to this notion of right patient, right target, right time. And I think genetics is not going to be the only answer there, but it's going to be a substantial component of any answer we come up with.
Yeah, what other information is going to be really helpful in that beyond genetics? If you want to start to, one of the things that I have always loved about genetics is the idea that you have this lifelong risk factor, and that could enable you to treat earlier, but you kind of need a quantitative biomarker beyond the genetics if it's neurofilament or noticing subtle tremors through a wearable device or something like that. I'd love if you could just speculate, or if you have data, then great, on what are the early biomarkers that we might be able to use to see this vision come to life.
Yeah, I think there's a ways to go for biomarkers in Parkinson's disease, but there's some evolving things. A lot of us are starting to apply proteomic biomarkers at scale, and I think combining that with genetics is going to be useful. You may have read of the synuclein seeding amplification assay as an assay that gives a readout for synuclein accumulation in individuals. So understanding where genetics fits into that is, are the genetic risk factors that we find, are they drivers of synuclein seeding? Are they a response to synuclein seeding? These things are all really important when you start to think about the temporal sequence of events and at what point you should go after each of those events therapeutically. So yeah, it's a rapidly changing field, and I think so interesting.
Yeah, so Sonya, as the program goes on, is there a way to layer these questions in? Like the synuclein seeding, you couldn't have known in 2020, right? And you have across the network I guess a way to build out core facilities in some way, right? To say we want to take samples from all centers or a subset of centers and do proteomics, or we want to capacity build locally, right? To enable people to do these within their country or geographies. Maybe you could talk a little bit about how you adapt to the times with this kind of global infrastructure.
Yeah, absolutely. And perhaps I can give the tangible example of GBA1, and Andy, feel free to jump in at any time. But at a very high level, essentially in the West African population, there's a GBA1 variant that increases your risk for Parkinson's disease. That variant less than 1% of Caucasian Northern Europeans would have it. So you wouldn't have found it otherwise. And what was really interesting about this variant was that it was in the intronic sequence. So not something you would have expected as a disease risk factor. So once we found that finding, we didn't just want to move on. We funneled some money to say, well, can we understand what the functional implications of this are? And so there was a study and a paper went out to understand that. But then we also wanted to understand that temporal component. How does this influence progression? What does this mean? And so now we want to follow up in that cohort and basically do a longitudinal study to understand what is going on in that particular population. So we're equipped to be nimble and responsive to those types of things. In terms of the alpha-synuclein seeding assay, we also invest heavily in PPMI, which was a program that MJFF spearheaded 10 years before we came onto the scene. But we have really believed in that program and helping grow that program to think through, can we do some more analysis, understand the genetics of those PPMI cohorts, understand who's at risk there to dive into this as well. Andy, is there anything you want to add to that?
Yeah, I just wanted to bounce back to the GBA1 example because I think this kind of, it's a really nice example of how GP2 operates, but also what we're trying to achieve. So the history of this is that we had 1,000 or so patients that had been collected from Lagos, from Nigeria and West Africa, and training is a really large component of GP2. So we were going through a training exercise. We were doing a genome-wide association study with roughly 1,000 affected individuals, and as you know, you wouldn't expect to see anything, right? So we were doing this online training using our data platform where the folks in my group were teaching individuals at University College London and in Lagos how to do genome-wide association study. And they found this hit completely completely by surprise. We were then able to show that it was a non-coding variant, which is, again, a surprise. That it was a branch point mutation that leads to decrease in protein and decrease in activity, again, by surprise. So you've got this training component, you've got this finding that would never have been found except for looking at an underrepresented population. It showed a new mechanism, a new risk mechanism, and it also highlights a population that would never have been chosen for a GBA-linked trial. Now there is a large population of African ancestry individuals who could be included in GBA trials and never would have been included in GBA trials before. So it's just a beautiful example of all of these things coming together.
Yeah, it really is. You can basically take every single lesson and run it through that one example. What do we know about you gave me a little bit that it decreases expression of the gene. Is it working? My understanding is the GBA variants that have been discovered in the mostly European ancestry populations tend to be a lot of missenses. Are they working in the same way, we think? Are they loss of function missenses, or might it tell us about GBA biology as well? Because there's maybe another layer to this.
Yeah, so you'd think we'd know this, right? After working in this space for a long time, there's a lot of controversy in the field. I would say most people think that risk for disease is imparted by decreased activity of glucocerebrosidase. So you have missense mutations, there are some stop mutations, some truncating mutations that all seem to impart risk for disease. The curious thing about this variant was that it caused an apparent increase in expression. So the risk allele caused an apparent increase in expression of GBA. And that's complex because GBA has a pseudogene right next to it. It's kind of hard to discern what's the pseudogene, what's the real gene. Yeah. But what the story is that the variant is in a branch point site, so it leads to inclusion of the intron, and you get an apparent increase in expression, but it's of non-functional transcripts. So what the net is that you get decreased protein. So it was a very cool finding, and we really went very rapidly from the genetic association to understanding the function in a matter of 3 or 4 months. It was great.
Where does therapeutics fit into the broader vision? Is there a way to use this program to help launch clinical trials? I would love to hear a little bit more about that.
Yeah, I would say in a couple of places. Um, so the first of all, the first part is knowledge transfer. All of our data is available. We make the data available through the Accelerating Medicines Partnership Program for Parkinson's disease, and there are many users from industry who access the data, analyze the data, take the results, and use that for the first kind of basic work. The other components are around understanding disease trajectories. So genotype-specific disease trajectories. You can imagine that you can power trials in a much more sophisticated way if you know about individualized disease trajectories. And then the piece that we started to think about more and more is, would we ever be able to use the structure of GP2, this collaborative network, to start to think about trial-ready cohorts?
Yep.
It's not the design of the study, but you can imagine that if there are a few 100,000 people enrolled in your study and lots of prospective studies going forward, that maybe the opportunities there to think about that in the future.
Sonya, anything to add to that?
Yeah, this is also an example of how GP2 pivoted a little bit. So essentially, once GP2 started, we have investigator meetings and investigators can give feedback. And a lot of investigators said we want return of results to be able to give back to our participant community. We recognize that this is research grade. Is there a way that we could have a CLIA certified result that we could give back? Some countries don't need the CLIA certification, some countries do. So there's also a geopolitical question there. So we thought, let's trial and pilot it out. So we worked with the Parkinson's Foundation through PD Generation, essentially, which is a program that provides return of results to individuals, either through a site or people can go online. And we thought, can we have them be a cohort within GP2 where we do whole genome sequencing? So it's not just the panel that they had originally been doing. So that way, as new results get added, we can then think about giving return of results as we get new discoveries as well. We had that infrastructure set up in the US, but let's also pilot to see, can they go outside of the US and think about that? And so we went into Latin America and South America as well. So we're also thinking about strategies of how to pilot or pressure test this in different parts of the world with different organizations as well.
Yeah. For longtime listeners of the podcast, we've had Jim Beck from Parkinson's Foundation on not once but twice historically, so you can go back and listen to. The first one was about the program as a whole, and we also discussed the expansion into Latin America. So it's really cool to see these macro level projects working together as well. Going back to the point you all made earlier, from the world that we existed in not too long ago, where there were really small hermit kingdoms of genetic data being hoarded, to one where you can actually have global collaboration like this, is pretty exciting, right?
Yeah,
absolutely.
It's incredible.
How do you think about the learnings from this program and how, bluntly, most other therapy areas don't have anywhere near this level of organization, and maybe also funding is a challenge, right? You have, I think in the paper you make the point of this major sustained decade plus long funding allows you to think big. So if we take the funding as a given for a second and say that, if you are able to find a funder that has this kind of long-term horizon, what are the learnings that people can take from this about how they might be able to apply this model in other places? And then we can talk about the funding point, which is probably really the problem of how do you actually find a funder with but it's a bit of both, I imagine. So maybe let's talk about how to set these kind of programs up and what you've learned along the way. Maybe I'll start with you for this one, Sonya.
Sure. I think one thing I learned that in retrospect seems obvious, but might not have been obvious when we started, is just thinking about who are the local champions and ambassadors for the cause that can then start their own cascade, right? The traditional model is you go into a place and you say, we're going to do amazing things. We're going to provide the resources to be able to analyze your data. You can come to our meetings. You can come get a PhD with us, which all sounds great on paper, but we're in two very different ecosystems. And so one thing we heard from the different countries or regions around the world was, we appreciate you, but we actually want to learn from our local and do more South-South collaborations and not always go to the US. We want to ensure that we have infrastructure to have training at home, and we're learning within our region because some of the geopolitics is going to be similar there. And I think that by by leveraging that model and also this train the trainer component, where when we go in to do a workshop, you identify a champion, and then that individual can then do the workshops afterwards, helps create this long-lasting, vibrant, scalable solution. And at the end of the day, a lot of the bioinformatics work, as long as they understand the condition of the disease, they could go someplace else. And so you could imagine, for example, another neurodegenerative disease coming in and leveraging whether it's the workshop infrastructure or the clinics that we've set up to say, we now want to be interested in disease X, and we can then introduce them to, here are the influencers that can connect you to that local community as you figure out who your champions want to be for your specific condition.
Andy, thoughts on that?
Yeah, no, I agree. We've been lucky enough to have the support to be able to invest a lot in building relationships and building capacity, skill capacity. So there's nothing like meeting face to face. That's for sure. So we have an annual investigators meeting. We have that on odd years, and then even years we have regional investigators meeting where we go and I spend time with local investigators. So I think last year we were in Casablanca, Cartagena, and Kuala Lumpur, each with about 150 people, local investigators, and there usually is a training workshop associated with that. That's a great way to connect. The training workshops, Sonya mentioned this, we have a train the trainer model where a group of 3 or 4 data scientists go out. They travel all over the world. They give data workshops. They identify 2 or 3 people that they think are great. And the next time they go, those people co-lead. Then the third time they lead. So you're kind of creating this
see one, do one, teach one kind of
a
model.
Exactly. That's it. Exactly. That's it. Exactly. I think we've done 15 of these now, something like that. There are 20 or so PhD studentships, and those include a program which I think has been really useful, which is sabbatical programs. So the PhD studentships are all at the local university. They don't register for a PhD in the US or in the UK or whatever. We select students who have a commitment to staying and building resources and building skills in their home country. But we give those students who are phenomenal the opportunity to come and spend 6 months somewhere else, whether that's in the global south or the global north, whatever. They get a chance to build connections, learn new things. And again, it's all about building this network of trusted individuals who are just driven to make advances in understanding disease and giving them some resources.
What question I meant to ask earlier, but what does the access to genetic testing look like across your network, US, EU, but also some of the parts of the world where our listeners probably would have no sense at all whether genetic testing is widely available or not? I'd love if you could just give a little bit of a landscape.
Yeah, it varies tremendously. And you've kind of hit on an issue here, or one of the early challenges that we came across, which is that actually the legal framework for genetics is different in each country, and for data sharing is different for each country. I'm sure that you've spoken a lot about GDPR and how to deal with GDPR. Right? That was a challenge that we had to get past. But the framework around return of results is different. So CLIA isn't a thing really outside of the US. So the regulations are just different. So in the same way that we have to think about how we deal with data in each country on a case-by-case basis, we also have to think about return of results and how we can deal with that on a case-by-case basis. As Sonya mentioned earlier, we've really piloted this by partnering with PD Generation to understand what this would look like in North America and in South America. We also have a return of results working group that is working on understanding how results could be returned in different parts of the world, with some emphasis in Southeast Asia and West Africa at the moment. So it's evolving and just very, very different country to country.
Is there any place in the world, though, where you'd say more than half of patients get tested by the healthcare system, or are we still far from that world. What I'm trying to get at is at some point, the evidence base it either will or it won't, but it probably will tip over into every Parkinson's patient should get tested, and the cost of testing matters, obviously. But I'm curious whether we're close to that. I don't even think we're anywhere close to that in the US or Europe, right?
No, no, we're not close to that anywhere. And I think this was part of the reason that the Parkinson's Foundation took on the PD Generation Initiative to try and inform us about the use and utility of genetic testing.
As we wrap here, I wanted to touch on the funding question. You mentioned that this program is supported by, I think, the Brin Foundation. Those who aren't familiar, Sergey Brin, one of the co-founders of Google, I believe his mother had Parkinson's disease, and I think he himself has been public about being a LRRK2 G2019S carrier. And I think he's donated more than $1 billion to Parkinson's research more broadly. So hard to understate the impact that he and his family foundation have had. When you think about applying this model more broadly, what advice do you have for other people who are looking at these kinds of programs? Is it possible to do this with smaller philanthropic efforts, with collaboration between industry and philanthropy? Part of the reason I bring up the industry question is because industry doesn't always have the 10-year horizon. They may have a 5-year horizon, but I'm just curious to get your takes on how to think about this. Where is it really philanthropists that can take that 10, 20-year view, or is there a way to put this together with short, medium, long term by building a little bit of a coalition?
Yeah, so as you had said at the outset, I'm the Chief Operating Officer for the Coalition for Aligning Science. So we're all about building coalitions. And it's really thinking through who are all the players in the space, irrespective of whether or not we have money or not. As Andy had alluded to, we have an amazing implementation partner through the Michael J. Fox Foundation. We could not have done half the work that we did if we did not leverage their infrastructure, both from a legal capacity, helping us navigate the geopolitics and contracts around the world, to the scientific staff that are also working with us. And so I think for any initiative, it's about mapping the landscape and figuring out how can we bring the organizations together. What we don't want to do is reinvent the wheel. And we also want to make sure we're checking our egos at the door. So in any space, I would always say, you don't know until you ask, what is the capacity that they can bring? And when you build those coalitions, you get stronger together, whether it's in industry, whether it's with other nonprofit funders. It's understanding where their strength is and where you are in the program, right? So industry might not have been as interested in GP2 when GP2 was just starting, but now that they are accessing the data, there could be potential opportunities. So it's also thinking about where you are in maturation for what you want to do, about which stakeholder might be more willing to come to the table.
That makes total sense. Andy, anything to add to that from your perspective?
No, just that my colleagues are very good at making sure my ego is checked at the door, usually by teasing me about something. So
I maybe on that vein, I want to close with an interesting story. I heard through the grapevine that you are so committed to GP2 that you have a GP2 tattooed on your arm. Is that true? And if so, can you tell us that story as we close out?
I don't know that I can tell you all of the story. I can tell you that I'm not the only person. I'm not the only lead in GP2 who has the tattoo. I can tell you that there were 3 of us, and there was some tequila involved, and then there was a tattoo following that in Barcelona. But we were very proud of ourselves, and it's a good story. And actually now we have these fake tattoos that we give away at the GP2 events, and they've become quite a collector's item. So I keep on trying to get Sonya to get one, but no luck so far.
My first response when Andy showed me the tattoo was that it was off-brand. So I said, if we're going to do this, we're going to do this right. We're going to have the comms team create these temporary tattoos that we'll pass on as well. Yeah.
That's great. We got our 3-year-old daughter little tattoos of her face for her 3-year-old birthday, and it was her favorite thing. So I'm a big fan of the novelty temporary tattoos.
Yeah.
That's so cool. Yeah.
I think that's an amazing note to end on. It's clear that you all have had an amazing impact already. It sounds like you've got 5 years plus to run on here. And Sonya, with your very thoughtful approach to how you build sustainability into these programs, I would be very surprised if this wasn't a program that just continued to compound and do amazing things for the field in general. So thank you both for your time today, but also for all the great work you've done for the Parkinson's community.
Thanks so much. It was lovely to chat with you today.
Great. And thanks everybody, as always, for listening, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.